Signatures of cognitive profiles in dementia, a cluster analysis using the Kaplan-Bayquest neurocognitive assessment. So we know there's significant variability in the trajectories of cognitive aging. So there's different types of dementia, variation within healthy aging from optimal to borderline impaired, and then different types of MCI. MCI being a stage of mild deficits that precedes dementia. There's recently been a trend in the clinical sphere towards diagnosing cognitive impairment earlier, as earlier diagnosis allows for better planning and management. And so there's a need now to better differentiate normal aging from MCI. In a previous study by Ladika, Wysick and colleagues, they performed a cluster analysis using the KBNA, and they found 3 clusters: a normal cluster, a mixed mild deficits cluster, and a globally impaired cluster. And they found that the normal cluster was best differentiated from the mixed mild deficits cluster using measures of verbal and visual long-term memory. So in our study, we sought to replicate these findings in order to validate them. So our research question was, which cognitive functions are first to diverge from normal during pathological aging processes? And to test this question, we performed another cluster analysis using the KBNA to confirm whether these measures of visual memory differentiate normal and mild deficit groups, which would suggest that these are the cognitive functions that are the first to diverge from normal during neurodegeneration. So our prediction was that visual and verbal memory will be the measures to differentiate normal aging from those with mild deficits. The participants were 210 patients at the Bayquest Center for Geriatric Care who underwent neuropsychological evaluation for diagnostic purposes. Our materials were cognitive screens, the Boston naming test, the trail making test, and the KBNA. And for our method, we used a retrospective chart analysis, so we went through patient files and extracted the relevant data. We found a 4-cluster solution with normal, mild deficits, moderate deficits, and globally impaired groups. We averaged the scores for each subtest to create the cognitive profile shown on the right. And then we examined the subtests on which the performance of the clusters diverged, which was all subtests. We then performed pairwise comparisons to see which clusters diverged on which subtests. So to begin with measures of memory in the verbal modality, we found that all pairwise comparisons were significant. So the normal group performed significantly better than the mild deficits group, and the mild deficits group performed significantly better than the moderate deficits group. Next, on to measures of visual memory. Once again, the normal group outperformed all of the other clusters. However, the mild deficits group and the moderate deficits group did not differ in their performance. Notably, the normal group performed significantly better by the greatest margin over the mild deficits group on these subtests. Finally, measures of working memory and verbal fluency. Here there was no difference between the normal and mild deficits group. However, there was a difference between the normal group and the moderate deficits group. As predicted, measures of verbal and visual memory differentiated the normal cluster from the mild deficits and moderate deficits clusters, which is consistent with previous findings. This suggests that these 2 cognitive functions may be the first to diverge from normal in pathological aging processes. In addition, the greatest difference was observed between the normal and mild deficits clusters in measures of visual memory specifically. So this may have a neurophysiological explanation, as Barbour and colleagues found that degeneration of the perirhinal cortex in monkeys is associated with impaired visual memory. And it's known that neurological changes associated with neurodegenerative diseases in humans often begin in the perirhinal cortex, which would explain why visual memory deficits were so pronounced in the mild deficits cluster. Future research could focus more on the trajectories of those with mild impairment. So it's unclear in our study if those in the moderate and mild clusters were on the same path, with the mild cluster simply being at an earlier stage, or if they were on completely different trajectories with a different likelihood of converting to dementia. And so longitudinal work could be useful to help establish the prognosis for different types of MCI. That concludes my presentation. Thank you for your time.